Michael Kyba from the University of Minnesota. Dr. Kyba will speak on discovering and testing therapeutics. Thank you, Dr. Kyba.
I want to say thank you to Dan and to June and the organizers for inviting me to talk about the research that we're doing in my laboratory. So what I thought that I would spend my 15 minutes on is to talk about, take you on a tour of a project that we undertook in my laboratory and published a couple of months ago. And several of you have actually emailed me about the results, and I thought it might be interesting for you to follow a project from conception through to the final publication. And so the basic idea is to try to discover a drug that might be useful in preventing the symptoms or blocking FSHD. So as you heard, FSHD is ultimately caused by this gene DUX4. And so just briefly, I'll give you an outline what DUX4 does to cells that has gotten the world very interested in its activity. Think about ways that we might block the activity of DUX4, and then finally tell you about this project that's been underway in my laboratory for a number of years. So what is it about DUX4 that makes it a very exciting candidate for FSHD? Well, the first aspect of DUX4 is really that it's right in the place where the mutation happens. So for a number of years, really, until about 5 years ago, most of the research on FSHD focused on these other genes that you'll see on the left that are different distances, some of them very distant, from the actual site where the mutation that causes the disease happens, whereas the DUX4 gene is actually right inside of those repeats that are altered by the mutation that causes FSHD. So it's in the right place genetically. Secondly, and this is an example that was of a study that we did a number of years ago, if you take the DUX4 gene and you put it into cells that don't normally express DUX4, it has very pathological effects on those cells. So on the left, you can see two panels, one of which is a picture of a cell. There are about 100 cells in that panel, and they're growing attached to a glass slide, and they're very healthy cells. And when the cells become unhealthy and die, they lift off and they round up and they float, and that's what you can see on the right. And the system that we set up in order to turn on DUX4 in these cells takes advantage of a drug called doxycycline. And we've set up a system where if we put doxycycline on these cells, we turn on DUX4. And it's a genetic system that we've set up. Cells don't normally express DUX4 when you treat them with doxycycline, and patients who take doxycycline would not turn on DUX4. But we've taken advantage of a genetic system that allows us to use this drug to turn on a transgene that we've stuck into the cells, and it kills them. In addition, we can see the DUX4 protein expressed in the cells within 2 hours. That's the red staining that you see on the upper right. And the shape of the cells changes dramatically. Within 16 hours, you can see that they stretch out before they lift off. And on the lower right, you can see panels that show the DUX4 protein. And so the upper panel shows that if we treat with different doses of doxycycline, those numbers 62.5 up to 1,000 are different amounts of dox. If we treat the cells with those different doses, we can titrate up, or we can express different levels of the DUX4 protein, from low levels to high levels. And it's the high levels of DUX4 that are killing the cells that you see in the upper left. You see they were treated with 500. The lower levels don't kill the cells. So an experiment that we did a long time ago was to test DUX4 against all of the other candidate genes that are in that area that other laboratories had been working on, to see how toxic are any of those genes using the exact same system. If you turn them on with doxycycline, and will they have an effect? And on the axis on this graph is the viability of the cells. So 1 means perfectly viable, and 0 means everything is dead. And so you can see that the 3 different doses from 0 up to 500 of DUX4 give a dose-dependent cell killing. The viability goes down. Whereas for the other genes, no change in viability was seen. So this is showing that DUX4 is very toxic, and at high levels it can kill the cells. However, at low levels it does something also very interesting. These are muscle cells mouse muscle cells that we can change. We can grow them as kind of like stem cells for muscle, and they grow and double inside the dish. But then we can also change the medium that they're growing in, and that will make them to differentiate and form muscle fibers right there in the dish. And so you can see the red staining in the middle series. Those are individual muscle fibers, and they're stained with an antibody that makes muscle fibers red. And that's at 0 doxycycline, so no DUX4 is being expressed in those cells. And as you titrate up, these are very, very low levels, but higher and higher, from 10 to 25 to 50. These are levels that will not kill the cells. But look what it does to the ability of those cells to make muscle. It's completely blocked their ability to make muscle, almost completely blocked by the time you get to 50 nanograms, a very low dose of DOX. So the fact that DUX4 can kill cells, the fact that it can prevent muscle stem cells from making muscle, even at very low levels, those are the reasons that the field is really excited about DUX4 in terms of the candidate causing FSHD. Now, it should be stated that there's no actually direct evidence for what DUX4 is doing in people with FSHD. It's very difficult to detect. And so all of this is extrapolated from cell culture models. Nevertheless, we're very excited about it. So how do you block DUX4? Well, we've heard in the beginning session and from Dr. Tapscott that one way to block DUX4 would be to prevent it from being turned on in the first place using drugs that alter epigenetics. And this is an extremely viable strategy that indeed it might lead to a clinical trial in the near future because some of these drugs are FDA approved. And the big advantage is that if you can block the activity, there's no DUX4, there's going to be no pathology. And then the disadvantage is that these drugs tend to have effects all through the genome, and they act on many genes, not just DUX4. And so it's quite possible, even likely, that there are going to be negative consequences of these types of drugs that it would be beneficial to avoid. And so the alternate strategy is to find a drug that directly targets the DUX4 protein itself and somehow inactivates it. So in distinction from strategy 1, the advantage is that this approach is extremely specific to DUX4. In theory, it might not affect any other protein in the cell, therefore it shouldn't lead to negative consequences for reasons that don't involve DUX4. The biggest disadvantage is that we don't really know if it's actually possible to target this protein and inactivate it. And it may be possible to do that in a dish, in a cell culture laboratory, but getting that molecule into the muscle cells of patients may be impossible. And so that would be the disadvantage of the second strategy. So nevertheless, we undertook an experiment to try and test whether we could find compounds that inhibit DUX4. And we didn't discover the magic bullet, but I think we learned something interesting about FSHD and something about some data that I think is suggestive of a type of clinical trial that ought to be done in the field. So this is the strategy that we're undertaking. So how do you find new molecules that will inhibit a protein? Well, you start with a chemical library. This is a set of compounds, thousands of them, each one is different, and they're kept in tiny little wells, and we can deliver those using robots into a cell culture dish and look to see what those compounds do to cells. So I showed you earlier that we can turn on DUX4 with this engineered system and kill the cells. In biomedical research, we call that type of experiment an assay. We can measure the activity of DUX4 by counting the number of live cells or dead cells or somewhere in between, a partial number of live cells, and we can get a readout that's quantitative. So you need these quantitative readouts. So we can assay thousands of chemicals to see if they prevent DUX4 from killing cells using a quantitative readout. Most compounds will have no effect. Some will inhibit cell death in this assay. And then we take those ones that inhibit cell death, those are called our hits. And there are, in a very large screen, you'll get hundreds of hits. Many of those hits will be protecting cells for reasons that have nothing to do with DUX4. For example, they might interfere with the genetic engineering that we did to turn on DUX4. That's a very common type of false positive in this type of screen. And so once you have your hits from this screen, you test all those hits in what are called secondary screens and identify ones that are specific to DUX4 versus ones that are non-specific. And then once you have identified ones that are specific to DUX4, since these compounds came to you on a library in very tiny quantities, we want to study them more deeply, so we have to purchase those compounds and do new experiments with those compounds using repurchased samples and try and investigate why is this compound preventing cells from dying when we turn on DUX4. So the screen that we did happened in stages. In initial stage, we tested a large series of compounds of known activity. And we identified 8 hits from this compound library of about 1,200 pharmacologically active compounds. But surprisingly, out of these 8 hits, 4 were actually poisons. And this was really surprising to us. And it just happens that the system that we set up is subject to the growth rate of the cells. The more rapidly cells are growing, the more sensitive they are to DUX4. The slower they're growing, the more resistant they are to DUX4. And so these toxic compounds that showed up as hits, what they were doing was they were slowing down the rate of cell growth. And that's shown on the lower right. If we grow cells in conditions where they grow more rapidly, which is the red bar, or more slowly, which is the white bar, and we turn on DUX4, the viability of the slow-growing cells is not affected nearly as much as the viability of the fast-growing cells. So this is one example of a false positive that we had to eliminate from our set of hits from the screen. So having done that smaller screen, we ramped it up to 44,000 random compounds. And what I'm showing you here is a subset of 10,000 of those. Most of those are shown up on the bottom of the graph as having very low viability. But there's a few, and we set a cutoff at around 60% survival that gave survival of cells in spite of DUX4. And I should say this is survival after only 24 hours. It's not that the cells were completely impervious to DUX4, but 24 hours later, 60% of them were still alive or more. And so from those 44,000 compounds, we took these hits, less than 1% of them. So you can see on a plate on the left, those are your 2 hits on that plate. We collected all the hits onto what we call cherry pick plates, a plate full of hits, compounds that we can use for secondary screens. And then we did a number of secondary screens, including the one for cell growth rate that I mentioned earlier. So one of the first things that we do is just retest the compounds to see if they're active. And so we have 1,200 hits, and at different concentrations, most of those, or many of those compounds, when we retested them, they protected. The viability was up in the 0.5 or higher range. But on the right of each of those graphs, you'll see lots of compounds that didn't repeat. And this is just what happens when you screen 44,000 compounds. You'll find things that, for just random statistical reasons, show up above the line. And we have to eliminate them by these repeat assays. And so here's an example of the ones that we're really excited about. So there were about 50 compounds, and they fell into particular structural classes. And some of the more common structural classes are shown above. Those are chemical structures, and the part that's highlighted in blue was common to a number of different compounds that showed up in that group. And below that is a graph that shows the activity of the compounds. Some of the compounds were toxic, so as you increase the amount, they actually improve things for a while, but then they make things worse because they begin to have toxic effects on the cells. So what we're showing you is the viability in this particular assay at the best concentration. And we've aligned the compounds from 1 being the most effective to 52 being the least effective. But they were all to some extent effective. So the next thing we wanted to know was, how are these compounds working? So we asked the question, do these compounds that protect cells from dying from DUX4, do they also protect cells from dying for other reasons? So we treated the cells in 6 different ways with 6 different cell-killing reagents, basically cell stressors. So you can see examples of those. One of the compounds causes DNA damage, another one causes calcium stress, oxidative stress, different types of stress to kill the cells. And the viability of the cells goes down, and in the presence of each of these 52 compounds, we're testing, does the viability come back up? And so on each graph, you can see a red line. If the compounds come above the red line, that means they're active in that assay. And so what's obvious from these graphs is that actually most of the DUX4 protective compounds were also protective against oxidative stress. but they were not protective against any of the other ways of killing cells. So that's very interesting. And then it turns out if you test other compounds that are generic antioxidants, they also protect cells from the toxic effects of DUX4 in this 24-hour assay. And so it looks like DUX4 expression, the screen has taught us that DUX4 expression leads to oxidative stress in this particular setup, which causes cell death. And we'd like to have a compound that inactivates DUX4, but other ways of getting a readout in this screen are compounds that prevent oxidative stress or protect cells from oxidative stress. And so these are the ones that we've mainly picked up in this screen. About 2/3 of the compounds did that. 1/3 of them did something else, and we don't know what that is. And there may be another pathway to cell death that's caused by DUX4, or these, among that set, may be something that directly interferes with the DUX4 protein. So that's the essential result of the study. And I wanted to just end with this question, which a number of people have asked me, is antioxidant therapy, because many of these compounds were antioxidants, is it a good idea in FSHD? And what I would suggest is that it's a good idea to test. It's a really good idea to test, and it would be nice to set up a clinical trial to follow the long-term consequences of antioxidant therapy. However, keep in mind that there's a difference between having a healthy amount of antioxidants and then overdosing yourself on antioxidants. So as typical on individuals, we're in a Western diet, and we're probably not receiving the appropriate amount of antioxidants through fruits and vegetables. Most of us are deficient in those. And so being deficient is probably a bad thing, and making sure that you're sufficient is probably a good thing. However, overdosing on antioxidants can have all kinds of negative consequences. So oxidative activity is important in your cells, and antioxidants aren't good for everything. There are cancer-killing cells that require oxidative activity to kill cancer. And so high-dose antioxidants could carry a lot of significant risks. And finally, I'll just end by thanking the individuals who did the work. Siho Choi, you can see, did the majority of this work, the fellow outlined with the arrow on the right, and a collaborator who's a medicinal chemist, Michael Walters. So thank you very much.
Thank you.